Hello everyone, I am Dr. B Ramya, Assistant Professor, PG and Research Department of Biochemistry, Auxilium College, Vellore. In this video session, we are going to discuss about the structure and functions of mitochondria. Through this video, students will able to learn the discovery of mitochondria, structure of mitochondria, composition and origin of mitochondria, and functions of mitochondria. Mitochondria are double membrane bound organelle known as the powerhouses of the cell due to their primary role in generating adenosine triphosphate, that is ATP, through cellular respiration. Their unique ultrastructure is perfectly adapted for efficient energy production and a variety of other critical cellular functions. Mitochondria are found in both plants and animal cells, absent in prokaryotes. Discovery of mitochondria. Mitochondria was first observed by Fleming and Kolliker in year 1882. The term mitochondria was introduced by Benda in the year 1898. Mitochondria may be filamentous or granular in shape. Structure of mitochondria. Mitochondria contains outer membrane space, forens, matrix, granule, ribosomes, mitochondrial DNA, ATP synthase, cristae. We will discuss the ultrastructure of mitochondria. Ultrastructure contains the following components. They are outer membrane, inner membrane space, inner membrane, mitochondrial matrix, mitochondrial DNA. Outer membrane of mitochondria is a smooth protective barrier which contains channel protein called forens, making it freely permeable to small molecules and ions. Inner membrane of mitochondria is the space between the outer and inner membrane is called perimitochondrial space. The chamber is filled with fluid or low viscosity and density. Inner membrane. This membrane is highly impermeable and selectively permeable, requiring special transport proteins for most molecules to cross. It is extensively folded into inward projections called cristae. The cristae is covered with small particles called F1 particles or elementary particles. Outer membrane of F1 particle is stalkless and inner membrane is stalked. Each F1 particle has a base, a stalk and a head. Mitochondrial matrix is the innermost compartment which is gel-like substances that contains a concentrated mixture of enzymes, mitochondrial DNA and ribosomes. Mitochondria contains one or more DNA called mitochondrial DNA. It's circular in shape and double-stranded. Composition of mitochondria. Mitochondria is composed of more than 70 enzymes and coenzymes, proteins, lipids, RNA, small amount of DNA, iron, copper, sulfur and some vitamins. Origin of mitochondria. The new mitochondria originated by the division of pre-existing mitochondria. Sometimes the mitochondria become elongated and broken into small pieces and each piece forms a new mitochondria. Functions of mitochondria. The predominant functions of mitochondria is the ATP production, that is the cellular respiration. The primary function is to convert nutrients like glucose and fatty acids into ATP using oxygen. The Krebs cycle and oxidative phosphorylation takes place in the mitochondrial matrix and inner membrane respectively, generating most of the cell's energy. Second function is the programmed cell death, that is apoptosis. Mitochondria play a central role in initiating the intrinsic pathway of apoptosis, leading to cell dismantling. Third one is calcium homeostasis. They help regulate the concentration of calcium ions in the cell, which is essential for cell signaling, muscle function and neurotransmitter release. Fourth one is heat production, that is thermogenesis. In certain specialized cells such as brown fat, mitochondria can generate heat and the process known as the non-shivering thermogenesis. Fifth one is metabolic regulation and synthesis. Mitochondria are involved in various metabolic pathways, including the synthesis of certain hormones like testosterone and estrogen, some amino acids, and heme groups for hemoglobin. Sixth one, cell growth and regulation. They influence cell differentiation, growth, and cell cycle, ensuring proper function and tissue health. Seventh one, semi-autonomous replication. Possessing their own unique circular DNA and ribosomes, mitochondria can synthesize some of their own proteins and replicate by binary fission, independently of the cell's nucleus. Eighth one is calcium accumulation. Calcium can be accumulated in mitochondria several hundred times than the normal. This process occurs during the formation of bones. Urea cycle. Initial step of urea cycle occurs in mitochondria. Thank you.